This part is the last part of chapter 12, part 4. In this part, I want to talk about neurotransmitters. The neurotransmitters are some chemicals which are synthesized by neuron and they are stored in the vesicles in the synaptic knob. When the action potential reach the synaptic knob, these vesicles start to release. And I told you that before releasing of this neurotransmitter, the calcium ion should come to the synaptic knob. Calcium ion should attach to the vesicles, and this combination can do exocytosis. When exocytosis takes place, they release in the synaptic cleft. They bind to their receptor in a target cell, and they trigger a physiologic response in the target cell. Approximately, we have 100 different types of neurotransmitters. The four major chemicals which we have for the neurotransmitters are listed here, and you can see them in this slide. The classification of neurotransmitter by their chemical structure are these. Some of them are acetylcholine, some of them are biogenic amines, some of them are amino acids, and some of them are neuropeptides. The structure of acetylcholine differs from other transmitters. The biogenic amine is the amino acid which is slightly modified to synthesize the transmitter. We have two different types of biogenic amine. One of them, is called catecholamines, like dopamine. They are made from tyrosine. And the other one is called indolamines, like serotonin, and they are made from histidine or tryptophan. The other one is amino acids. The amino acids include common transmitter glutamate, glycine, and GABA. And the last one is called neuropeptide. The neuropeptide are chains of amino acid, 2 to 40 amino acids, and they have substance P and endorphins in their structure. Neurotransmitter also classified by their functions. By their functions, they may be, we can classify them, by their effect or by their action. By their effect, some of them are excitatory. They can make excitatory postsynaptic potential, and I hope you remember what is the meaning of excitatory postsynaptic potential. It means that they can open the sodium channel, sodium ion come into the cell, and inside our cell become more and more positive. Some of the neurotransmitters are inhibitory neurotransmitter. It means that they can make IPSP. The IPSP can happen by the opening of potassium channels or chloride channels. If the chloride ion come to our cell or potassium ion leave our cell, inside the cell become more negative and IPSP take place. We can classify the neurotransmitter by their action too. Some of the neurotransmitter can work directly. It means that the neurotransmitter directly bind to the receptor of the target cell and make opening of the ion channels. Some of them are working indirectly. The neurotransmitter bind to the receptor that activates the second messenger. So we need something in between, the neurotransmitter and the channel which become open. This part which is located in between is called G protein. So we have different types of neurotransmitters based on their function. In this slide, I want to talk about the function of the acetylcholine. The acetylcholine is best characterized neurotransmitter. It's used in the peripheral nervous system for stimulation of skeletal muscle and in the central nervous system to increase arousal. The acetylcholine is synthesized from acetate and choline, and it is stored in the synaptic vesicle. Action potential triggers its release to the synaptic cleft. Some acetylcholine can attach to the postsynaptic receptor, and the other acetylcholines, which are not attached, are cleared from the cleft by being broken down to acetate and choline by the enzyme which we call it acetylcholine esterase. So the acetylcholine esterase can break down the acetylcholine to acetate and choline. Acetate and choline are taken up by perisynaptic cell, and our cell can do recycling. Again, it can make acetylcholine. And the effect on the target cell are different. Sometimes it can activate the nicotinic receptor, sometimes it can activate the muscarinic receptor, which I will talk about them in chapter 15. We have three different ways for removal of neurotransmitter from the synaptic cleft. One of them is function of different types of enzyme, like acetylcholine esterase. The other one is reuptake. The perisynaptic transporter might import neurotransmitter inside the cell, which we call it reuptake. And sometimes some neurotransmitter can do diffusion, and they move from the synaptic cleft to the other part, and after that they become reabsorbed by the glial cell. Some drugs have their effect by influencing transmitter removal. For example, selective serotonin reuptake inhibitor can depression. Or galantamine hydrobromide is a drug which we can use it for treatment of Alzheimer's disease, and it can work as the acetylcholine inhibitor. It doesn't let the acetylcholine esterase work on our acetylcholine and break it down. So by this function, it can do treatment of the Alzheimer's disease. Here you can see the function of acetylcholine. As you see here, the acetylcholine, the nerve signal can come to the end of our axon knob. It helps the calcium ion to come inside the cell and then attach to the vesicles which contain neurotransmitter, and the acetylcholine is due exocytosis and come to the synaptic cleft. Sometimes it can work directly, as you see here, on our receptor, and sometimes it can work indirectly, and here we need one secondary messenger here. And after that, the acetylcholine can broken down by the acetylcholine esterase in the synaptic cleft. And we have choline and acetate, which come back to our cell, and our cell can do recycling and use them for the second time. The other type of chemical which we have in neurophysiology is called neuromodulator. The neuromodulator are the chemicals which can alter the response of local neurons. Some of the neuromodulators can do facilitation, and some of them can do inhibition. The neuromodulator, which can do facilitation, can cause greater response in the postsynaptic neuron. They may increase the amount of neurotransmitter in the cleft or the number of postsynaptic receptors. And the neuromodulator, which can do inhibition, can cause weaker response for us. They may decrease the amount of neurotransmitter in the cleft or the number of postsynaptic receptors. So there are some chemicals which can alter, which can change the response of our neurons locally. In this slide, you can see two different types of neuromodulator. One of them is nitric oxide. It can be a neurotransmitter or a neuromodulator. It's a nonpolar gas, and the lifetime of this nitric oxide is very short. It's made and released by the postsynaptic neuron in the brain, and it can increase the memory. It has one other effect in the peripheral nervous system, which you will learn about them in 2060. It can help our blood vessel dilation and decrease the blood pressure. The other type of neuromodulator is called endocannabinoids. These endocannabinoids are influences same receptor which marijuana does. It's very small and non-polar molecule. It's made and released by postsynaptic neuron, and it has the effect on the perisynaptic neurotransmitter release. It can influence the memory, it can influence the appetite, it can suppress nausea. These are the neuromodulators which we have in the central and the peripheral nervous system. Now you're ready to answer these questions. This is the end of chapter 12.